All right, in this amazing video, I am going to talk about plant growth and storage. Um, we're going to talk about, yeah, let's just move to the next slide because I think I have my bullet points here. All right, so I'm first going to talk about plant growth on both the cellular level and the whole plant level, and then talk a little bit about carbon storage. And then this will kind of introduce you to the concepts that I think you'll need to then read the rest of the chapter or part of the rest of the chapter on your own, and then we'll read some papers related to these topics. So let's first talk about cell growth. Um, so, we do talk a lot about individual cells in this class, and sometimes, I don't know, maybe that can seem like too small of a scale. But in this case, I think it's really cool to think a little bit about cell growth and how it really dictates, whole plant growth. And that's kind of like a basic concept, but it really does dictate how fast a plant can actually grow. So the rate of cell division and the rate of cell expansion are the two factors that really control tissue growth. So if a plant can divide its cells really quickly and expand those to the size it needs, then you're going to have a plant that grows fast because it's going to be able to produce more cells and produce the tissues faster, and result in fast growth. So it's really, it is occurring at that cellular level. So from a cell division standpoint, we don't want to get into too many details, but I think what's important and interesting is that there's some kind of pre-programmed size that a cell will reach before it divides. So once a cell hits whatever size it's going to get to, then that triggers it to divide. So anything that can speed up the process or slow down the process of a cell getting to that size is then going to have an impact on the rate of cell division. Now, from a cell expansion standpoint, there's two things that control how fast and how much a cell expands as it grows. So one is the cell wall rigidity. So cell walls of mature plants, those are fairly rigid structures. And so, when we talked about when a cell is filled with water, that water puts pressure outward on the cell wall. And then the cell wall pushes, is somewhat rigid and puts pressure back on the contents of the cell. So if that's really rigid, then it's going to take a lot of turgor for the cell to expand as it's growing. Whereas if it's very elastic, it'll take less turgor to cause that cell to expand. And so that's generally what happens during growth, is that the cell walls are really elastic. So it doesn't take a lot of turgor to cause a big, response in the expansion of the cell. And then after the cell reaches its final size, then it will become more rigid. So the plant can fairly quickly change how elastic or rigid the cell wall is. Um, and this slide just kind of reinforces some of the stuff from the other slide. But thinking about the elasticity of that cell wall, there is a family of proteins called expansins, and so they are most of them, well, as far as I know, they're all a type of enzyme that causes the microfibrils of the cell walls to relax and then allows them to be more elastic and expand. And so the cell wall has, that's what I should have had a picture on, not my silly drawing of a circle. But a cell is made up of all these little microtubules or fibrils that they're just like little strands that are intertwined on the cell wall. And so these expansins cause those microfibrils to be really elastic so they can stretch. And once they're stretched out and the cell reaches its full size, then those expansins are deregulated and those cells can become, or the microfibrils will become more rigid, and kind of that's the final size and shape of the cell. You often see low pH values um, corresponding with the production of these expansins, because the expansins are much more active and low pH. So that's what you see in areas of the plant where there's active cell growth. We have lower pH and we have lots of these expansins, and that's to make those cell walls elastic. And then the turgor pressure, so a situation where plants have plenty of water to develop high turgor pressure, that's going to lead to faster cell growth, because those cells are going to expand faster, reach that size where they're going to divide, and then continue that process on. So during periods of like high soil moisture or abundant soil moisture, cell division is going to happen relatively rapidly. And then when plants are exposed to drier conditions, then it's harder for them to generate a high turgor pressure. And so that's going to slow the growth rate of the cells. It also, I said a couple minutes ago that cells like have a certain size that they attain before they trigger their cell division, but that size isn't constant for all species or all times. During dry periods, the cells tend to be smaller, and so that what triggers cell division is a smaller cell in a droughted plant compared to a well-watered plant. But anyway, being able to generate that turgor, which these orange arrows are representing the pressure of the water or the fluid contents in the cell outward on that cell, that's going to increase how rapidly those cells expand and grow. Okay, so that's all cellular level processes. The area of the plant where cells are actively dividing and expanding are found in the meristem tissues of the plant. And those can be in lots of different places on the plant. But the two main areas we think of are the apical meristem, which would be at the top kind of the plant, or the apex of the plant, and then on the tips of the roots. And so we've got a couple of stained images. These are cross sections of plant tissue. Here the red is staining the meristem area, and then down here it's a little harder to tell from the stain, but you can kind of see this. It almost looks like a root within a root. This area here that I'm highlighting is the meristem zone, where cells are actively dividing. So down here, cells would be dividing and pushing outward, and then pushing outward towards the side as well. The reason I point out the meristem is because the size of the meristem also has a big impact on the growth rates of plants. So not only how fast the cells divide and how fast they expand impact plant growth, but also how many cells you have to do the dividing and expanding, right? That also impacts growth rate. So if you've got a lot of meristem, you probably have a lot of cells that are actively dividing and expanding, and so that'll also lead to faster growth. So the size or the volume of the meristem, as well as that division and expansion. Okay, enough about cellular level processes. Let's talk a little bit about what's going on at the whole plant. And again, this is just kind of an introduction. As we read in the book, there's going to be a lot more about kind of allocation patterns in plants and how plants adjust what tissues they're allocating biomass to. And so this is just to kind of get us started in that topic. Okay, so first, I want to talk about relative growth rate. So we're going to talk, or in the within this chapter, and we talked about it a little bit with the nutrients, we talked about relative growth rate. And relative growth rate is, okay, let me just go by my bullet points and not jump ahead. So growth rate by itself is largely impacted by plant size. So larger plants have more leaf area for photosynthesis, and so they're going to grow faster. If we're kind of presenting, like growth would be presented on like grams of new biomass per day, right? So if we were to compare plant A and plant B has got more biomass, more leaf area, to do photosynthesis. So it's going to accumulate more carbon per day, assuming photosynthetic rates are pretty similar. So plant B is probably going to have a faster growth rate than plant A. But it's only because it's larger. Okay, and it might be because it's older, and that's why it's larger. What becomes most interesting to us, at least in most situations, is the relative growth rate. So that's the growth rate, the new growth that's produced relative to how big that plant is. So relative growth rate here, as I've defined it, is new growth divided by existing biomass, or the new biomass production divided by existing biomass. And this is in typically, I forgot one unit here, it's in milligrams of new biomass per gram of existing biomass per day, or per year maybe, per unit time should be in there. So that's why relative growth rate is really important, is because we can compare small plants to large plants by calculating relative growth rate. So we're going to use relative growth rate a lot. Oh boy. All right, I've got to change this. Reishi on. Uh-oh. I'm having trouble with my mouse. I think my batteries are getting low. Oh, I think they died. Oh, shoot. Okay. Whew. That was dramatic. The other thing to think about a bit now, and again, we're going to talk about this more later, but is just kind of how much of the plant is allocated to leaves. And we already talked about this with relative growth rate to some degree, but this is just a way of quantifying that. So a large proportion of leaves relative to other tissues is going to typically result in faster growth rate. So the way we quantify this is with the leaf area ratio, or LAR, which is meters squared of leaf area divided by plant biomass. And the way we're going to calculate this is the specific leaf area we're going to talk about this more in just a second, but it's meters squared of leaf area divided by grams of leaves times the leaf mass ratio, which is the grams of leaf area divided by grams of total biomass. So this is just a fraction. It's the fraction of leaves relative to total biomass. And this is the area of leaves per biomass. And if you multiply these two together, you get meters squared per plant biomass. So large LAR values means a plant is allocating or has allocated a lot of its biomass to producing leaf area. More leaf area means more photosynthesis and typically faster growth. So the plant A and plant B are similar to the ones before. Plant A, relatively small, a small amount of leaf area relative to the woody tissue. Plant B has a much smaller stem, so smaller amount of biomass allocated to woody tissue, but much more biomass allocated to leaf area. So this particular plant is going to grow really, really fast because it's allocating so much to the leaf area, and that leaf area then is accumulating more carbon. Of course, we might end up with plants that are slightly unrealistic as far as like growing on the landscape, but that's okay. All right, let's talk briefly about specific leaf area because this is going to come up a little bit in the chapter as well. So specific leaf area, if you're not familiar with this term yet, or this plant trait, man, it's used all the time, and it's overused in my opinion, but it is super valuable. So this tells us something about how much a plant is investing in its leaves relative to the leaf area that they're going to be able to accumulate carbon with. So it's the meters squared of leaf area divided by mass. So if you have a large SLA value, that means you have a lot of leaf area relative to how much biomass was invested in those leaves. So that kind of translates to larger SLA values being like, or characterizing what we would call cheaper leaves. So they didn't take as much to build. They were a small investment, and that allows plants to achieve a high leaf area ratio. So lots of leaf area relative to total biomass. But there is a trade-off. Those leaves, because they're cheaper, they tend to be a little wimpier, for lack of a better word. They can be damaged easily. Herbivores like those leaves better, and so they tend to be damaged from herbivory or wind or hail or other kind of disturbances. So there is a trade-off, but when we're thinking about fast growth rates, plants that grow really fast tend to build leaves that have a high SLA, or these relatively cheap leaves. Okay, the point of those last few slides is to get you thinking about like kind of what's going on, not only like during photosynthesis at the leaf level, but then how plants are using that carbon to grow and construct tissues, because that can have even a bigger impact on growth rate than just photosynthetic rates. So you could have a plant with kind of a slow photosynthetic rate, but if it's allocating a lot of biomass to its leaves, as a whole, it might grow faster than some other plant. Now, of course, there's trade-offs with that, but just thinking about growth rate right now, that's kind of a realistic scenario. So I hate to admit this, but it's not all about leaf level photosynthetic rates. There are other important things to consider at the whole, excuse me, the whole plant level. Okay, let's talk a little bit about carbon storage. The three main forms of carbon storage are the sucrose, fructans, and starch. So sucrose and fructans are water soluble, and because they're water soluble, they can be transported in the phloem. So these are smaller forms of carbohydrates that are moved back and forth within the plant. And then starch is a much larger molecule. So starch is essentially a bunch of sucrose and fructans put together to make a really large molecule, and it's actually water insoluble. The advantage of that is that it's inert, so it's not going to react with other molecules, and it's not going to change the osmotic potential of cells, or of the fluid in cells. So sucrose and fructans will impact the osmotic potential, starch will not. The downside is that it can't be transported in the phloem. So if you've got starch somewhere and you want that carbon somewhere else, you have to break the starch down into sucrose or fructans or some other small molecule, transport it, and then you could use it elsewhere. So starch is typically formed in the location where it's going to be stored, that last bullet point there. So the amount of carbon storage, something I wanted to, I guess an important point to point out here is that there's two things to consider when we're thinking about how much carbon or how much carbohydrates a plant is storing. There's the two factors are the volume of storage tissue. So if a plant has a large volume, it has a large capacity to store carbohydrates. Of course, that comes at a cost because that plant has to produce biomass to house those carbohydrates. But then also the concentration of the carbohydrates in that storage volume. That can vary between species or over time. And so in order to know how much carbon a plant has access to, stored carbon a plant has access to, you can't just measure the concentration, which is commonly done because it's relatively easy. We might sample some roots or part of a stem and say, oh, look, the concentration of this species is higher than that species. But if we don't know how much volume each of those species has to store carbohydrates, we might miscalculate or misjudge how much carbon those plants have stored. So just keep that in mind because the volume of storage is less frequently measured. Oftentimes people account for it in the discussion section of a paper or something, but it's not measured as much. And it's not a criticism because it's really hard to measure that, but it's just something that you have to keep in mind as you're reading these papers and interpreting that data. I don't know if we need to go over this, but I just gave a couple of scenarios down here. So let's assume plant A and B, these beautiful plants over here, they store most of their carbohydrates in their stem. So in the orange rectangle. So in scenario one, if both plant A and B have the same concentration in their stems, then plant B is going to have a lot more carbon stored, right? Because it's got a bigger stem, so it has more carbon, and if it needs it, it's got it there. And so that would be a common way for people to characterize plant A and B, is just taking a section of their stem and measuring concentrations. But then we would need to measure the volume as well. In scenario two, if plant B has a lower concentration of carbohydrates in its stem, it might still have just as much carbon because it's got more volume. So kind of the same thing, but two different scenarios that can arise. Hopefully that reinforces that point. Okay, briefly about the location of carbon storage. So carbon storage can happen in pretty much any living tissue. I guess I should have said living tissue there, but hopefully that was what you were assuming. Roots are a common place for carbon to be stored, especially large tap roots. In the alpine, I was trying to find a good picture of an alpine plant, because a lot of times you see these alpine plants and it will be, a plant that's just a few inches tall and a few leaves, and then it has this like giant taproot that's a couple meters deep and might be a couple inches in diameter. And so alpine plants in general are known for storing lots of carbohydrates. So anyway, that gives you a visual, but that's one common place that carbohydrates are stored. The stems of trees are places where there's lots of carbon stored as well. And then leaves can store carbon as well, but it's usually for shorter time periods. There's not as much volume, storage volume in the cells of leaves, because as you know, a lot of those cells are, have all of the photosynthetic machinery, and so there's not a lot of room left over for storage. So typically leaves are kind of a short-term storage for carbon, and then the woody tissue or the roots are more of a long-term storage. Carbon storage is also happening on different time scales, which I kind of alluded to there. So on the short term, plants are accumulating carbohydrates in their leaves during the day as they're photosynthesizing, and then they tend to export them at night. And so some of these figures here just show that diurnal pattern. This was just one of the first graphs I found. It's kind of interesting. This is the wild type Arabidopsis in the closed black symbols. And then the open squares here is a mutant that can't produce starch. So obviously, since it can't produce starch, there's no change in the concentration of starch, which is on the y-axis, over the course of the day, which is here on the x-axis from 0 to 24 hours. But what you see is during the daytime, these leaves are accumulating starch until it gets dark, and then it's breaking that starch back down and exporting it to other parts of the plant. So that's kind of what happens on a relatively short time scale in the leaves. We can ignore this figure to the right. It's not relevant, or not important, I should say. On the long term, plants can put carbohydrates into roots and stems, and I think there's probably most trees and maybe most plants, most perennial plants, they put that carbon away, and there's some carbon they probably never touch. I certainly believe that's true with trees. I'm not sure about grasses, but they're just constantly putting carbon into storage, kind of like as a fail-safe, right? If they run into a situation where they need lots of carbon, they've got it. But it seems like most individuals don't ever hit that dire situation where they need to deplete all of their carbon stores. And so I think there's probably carbon that's accumulated in the first couple years of growth that never gets touched. So that's really, really long-term storage compared to the short-term storage. Okay. Um, I feel like the carbon storage piece, it's really important, really interesting from a management perspective. Certainly we think about it in trees and shrubs and other species like that or other functional groups because it's important for the survival and regrowth of plants. But where I feel like it's been used most in management is in rangeland ecosystems. Like if you look in the 60s, but especially in the 70s and 80s in the range literature, man, everybody was measuring carbohydrate levels of every single species that was important for forage. And what they were doing was looking for changes in the concentration of carbohydrates in a plant, and then um, trying to see if the timing of grazing was important for a plant to be able to recover its carbohydrate stores. So just as an example, this graph here is I think it's for crested wheatgrass or Russian rye. I can't remember which one now. And so you can see on the x-axis we have from September of one year to November of the next year. And then on the y-axis, we have the total available carbohydrates in the roots and crown tissue of these plants. And what you see is that in September, it's relatively high and it gets depleted through the spring. And you can see in kind of April, we're at the lowest level. And this would be right when those plants are resprouting. So they've survived the winter time. They've used some of that carbon to keep all of their cells alive. They've probably supported the growth of new leaves or produced the new leaves from that stored carbon. And then they can replenish that carbon. And you can see it goes back up and we hit a maximum sometime later in the summer, July, August period. And then you can see it starts to decline again. So from a management perspective, managers have used this information in two ways. One, if you want to get rid of a plant on the landscape, you can graze or browse it right here when it's hit its minimum, because that's going to make it harder for that plant to recover. And over time, if that's repeated, that's going to have big impacts on the growth of that plant, and it might become less abundant on the landscape. On the flip side, if you want a particular plant to be abundant on the landscape, you don't want to graze it or browse it in this early spring. You want to wait till those that carbon concentration has increased. And that's like on the shortgrass steppe, which is kind of northeast of town here. That's what they do on most of those pastures. They don't put the cattle out till like April or May, after those plants have started recovering. So they make sure not to hit them when they're kind of at their low point. So again, there's other examples of carbohydrates being used for management, but I think that's a really cool example, and it's used quite extensively from that perspective. Okay, so using stored carbon, very briefly, and I feel like I've alluded to pretty much all of these things that I pointed out here, but plants can break down those stored carbohydrates into smaller sugars and transport them to wherever they need in the plant. And usually those needs are one of two things. It's going to grow new biomass, and so it needs some carbon to produce that biomass, or it needs energy. So remember, the plant can take those sugars and break them down and release ATP or produce ATP that then it can use to drive physiological processes. So wherever that energy is needed or the biomass is needed, that stored carbon can be translocated to that area. And so as long as the phloem is working, plants can use that stored carbon to kind of regrow or survive or, produce new biomass. From a drought mortality perspective, that's of course been more prevalent in the literature lately, and kind of what kills plants during drought. What they've found is, plants don't usually run out of carbon, but they might not be able to move it. So remember when we talked about phloem transport, I think we talked about this a little bit, but to move sugars or carbon from one place to another, you have to create a positive hydrostatic gradient, right? So you load your sugars in the phloem in one place, and you need water to be able to fill those cells and create high turgor pressure to push that somewhere else. So if you don't have enough water to actually like fill those cells and create that pressure, you're not going to be able to use your stored carbohydrates. So in that situation, a plant can't use the stored carbon. But otherwise, that stored carbon can be used and moved throughout the plant. Okay, that's all I've got for this. There is a short quiz associated with the lecture, so make sure you take that before the due date. And that's all for today.